I'm Diane Alston, and I'm going to talk with you about some of the primary insect pests that we deal with in our orchards. So I'm just going to build upon some of the baseline information that Marion gave you about degree days and timing and mating disruption and how things work. All right, so now it's not advancing. So hard with these things where you rely on so much technology.
Resume slideshow. Go now. See
okay, now it is. Thank you. So the three primary insect pests that we deal with in orchards in the Intermountain West. In apple, our top one is codling moth, and Marion talked quite a bit about this one already, and I'm going to go into more depth about its management. Peach twig borer, in peach, apricot, and nectarine. And then western cherry fruit fly is our key pest in cherry, insect pest in cherry. I'm also going to briefly give you some updates on two invasive insects that are of concern to our orchard production, the brown marmorated stink bug and spotted winged Drosophila. So we'll talk briefly about those. And my goals of what I want to share with you today as we go through these different insects is to give you some understanding of their biology, because if you understand a bit more about their life cycles and where they are at different times of the year, that really helps you get a better handle on how to manage them. And then about monitoring, Marion talked about this in a general sense, and I'll give you a little more detail for these specific pests. And then, of course, management is the biggie, and IPM strategy. So what options do we have to manage these pests in the best way possible? And then refer a lot to information resources, which are pretty much all online. So let's start off with codling moth. So this is our key pest of apple and pear. Some things you most people, fully recognize the picture of the worm and the apple and are very familiar with that. But some things that you may not know about its biology that could help you is that it actually does overwinter as a mature larva inside a webbed cocoon underneath protected places. So in a natural setting, it likes to be under the rough bark of apple trees or any other kind of tree that's nearby. But also will take advantage of any kind of structure. So if you have wooden bins or other things that you leave out in the orchard, or there's other buildings or any kind of structure like that, it'll take advantage of any of those places. But that's where it spends the winter, and that's where it is right now. And then in the spring, when it gets warm enough, talk about degree days, when we reach that threshold for development, out will come the adult stage, so male and female moths. Those fly, mate, then lay eggs, and they lay eggs on the fruit surface itself. So it's beginning in the first generation in the spring, late spring, it's little tiny fruit, and then as the season goes on, the fruit's getting larger, and it lays those singly on the fruit itself or on eggs that are on leaves that are in the cluster where the fruit are located. And in a survey that Mary and I did this past summer, we found quite a few eggs laid on leaves. So we were focused on fruit, but then started looking at the leaves near the fruit and found a lot of the eggs laid on leaves as well. So one thing that's important to know about codling moth is that for this insect, that dormant oil spray is not effective. And the reason is at the time that you would be applying that, right when the buds are starting to open, starting to see that first little leaf tissue showing up, the codling moth is still in that cocoon underneath protected bark. And even if you coat the tree with an oil spray, you're not going to kill the codling moth larva inside that cocoon. It's just too protected in there. And so this is one of our pests where the dormant oil is not a tool that you use. Unfortunately, we wish it was, but it's not the case. So when that adult moth emerges, right about apple bloom time is when they become active. I mentioned they lay their eggs on the fruit and the nearby leaves. And then once that little egg hatches, it's very small. They're hard to see. Mary and I got quite good at looking for these little tiny eggs this past summer out in the apple orchards. A little tiny first instar, that's the neonate, the very first larval stage, caterpillar stage, hatches out. And within 24 hours, that little bugger chews into the fruit. And so that's a very narrow window of time between when that egg is there, that egg hatches, and then that larva is inside the fruit. And that creates a great challenge for the management of this insect, because that window of time of when you have access to do anything about it, to keep it out of the apple or pear, is fairly short. And so that's why timing is also critical with this insect. So at the bottom there, some of the photos I have, it shows one of the eggs. It's a little hard to see, but it's this collapsed white structure that we had circled on an apple when we were out sampling. And then you can see that when that larva hatched, it crawled just a short distance, and then that little brown area where it tunneled into the fruit is just right on the edge of the black Sharpie marker circle that we made. So you can see when that egg hatched, the larva went right there, and whammo, it's in. Also, if you didn't know this, the larvae actually like to feed on the seeds of the apple and pear, and that's their goal. They're tunneling into that fruit to get down to where those seeds are developing, because that's where they want to feed. And that's why they'll cause so much damage in the flesh. They're not just staying near the surface, they're tunneling right down to the center. Okay, so I've got some questions about management, but I would like to, I'm going to talk about these, so just hold on, you guys. You're jumping, you're putting the cart before the horse, and I'll make sure I come back and talk about all these, all right? Thanks. This is supposed to be an introductory class, so I want to go through the basics and talk about the biology of the insect so that you can understand how we build the management upon that. Okay, so Marion mentioned that pheromones are the key for monitoring this insect, and that is true, and she talked about how the pheromone plume in the male focuses in on that. And we use a trap to set that biofix. So the earlier question we had about biofix setting, if you're using a trap to set biofix, then our model is all based on when from that biofix point. And I'll show you in the next slide how a kind of typical flight pattern. And so that is your biofix. Doesn't matter whether you started the degree days on January 1 or March 1 to get your timing to put out the trap. Once you've set that first biofix or first moth activity, that's when you then start all the degree day models from, then to calculate when it's time, when eggs will be hatching, when larvae will be getting into fruit in the next generation, and so on. So if you have a solid biofix, then you're all set. But as Marion mentioned, we're having more challenges with setting biofixes for a number of reasons. One of them is that we are using mating disruption, which is a great tool, but what's happened now is it's lowered the populations in a lot of the apple and pear orchards, and so you put out a trap to set biofix and you don't catch anything. And so then now we've been recommending that growers put a trap in a place they know is not being disrupted with pheromones, so maybe a neighboring orchard or a backyard tree, something like that. But then you're not getting the biofix for your orchard, so that creates some challenges. So a question was brought up with Marion's talk about this another method they've been using up in Washington State, or for the latitude that their orchards are at there on the eastern slope in Washington, they've been using 175 degree days to just say that's when we start our, that's our biofix, and then we start calculating from there. And so for different locations, they find out what date, calendar date, that 175 degree days is for them. So Marion and I started a project last summer to look at whether this would be a suitable approach for Utah. Our latitudes are lower than Washington and our elevation is higher. And so we're using a formula that's based on latitude and elevation. And so in this first summer, 2016, we found a pretty good fit for our northern Utah sites. So anything from southern Utah County up to Box Elder County. We found that using actually a March 1 start date gave us a pretty good prediction of when that trap catch, that biofix would occur. When we used January 1, we actually had a lot more variation in the data, and it could be anywhere from -10 to +5 days. That's too big of a range. That's not accurate enough. But with the March 1 start date, using the latitude and elevation of the site, we were able to be within a couple of days of when the biofix actually occurred. So year 1 data looks pretty promising that that model may work for northern Utah. Unfortunately, it did not work for southern Utah. We had sites down in New Harmony and Cedar City, and they did not get, the prediction was very poor down there. So we're still going to have to figure out what's going on there. And we're repeating this study in 2017, and we'll have a bigger data set. And then from there, we can make some more permanent recommendations of what we're going to recommend here in Utah. And I will talk about that in much more detail at the Utah State Horticultural Association meeting, which will be here in this room January 20th, 21st, something in there, I forget the exact dates. But so come to that if you want to hear more about that research. So this is what a typical collie moth flight pattern looks like if you're not using insecticides, where that first little bump in the red line there is the first generation, smaller. And then if you don't do anything to manage collie moth, you get a much bigger second generation. And then now this is a graph that I made for a class many years ago, and now we get a significant third generation also as our temperatures have warmed. And so you were looking at the number of moths that you're catching in a trap per night. And then you can see down in the lower left-hand corner, there's a little biofix arrow. So that was when you had your first moth catch at your biofix. And then you see some other green arrows that say targets for control. And so you see that for first, second, and third generation, I've put up there the degree day timing that matches up when eggs start to hatch. So based on when those moths first fly, it takes about 220 degree days for the mating to happen, females to mature their eggs are laid, eggs hatch. So that 220 to 250 degree day timing corresponds to about 1 to 3% egg hatch for the first generation. And then we have respective numbers for second and third generation. And all that is related back to that very first biofix state of when collie moth became active that year. So that's why biofix is such an important entity, because it's a tool that we use to time the whole season. Now I'm going to come back to more about that timing and degree days and showing you our traps website and how in Marion's pest advisory she comes up with those dates to recommend to you. But I'm going to diverge just for a little bit and get into mating disruption, because I'm trying to deal with this a little bit chronologically. So Marion introduced mating disruption. It's important to place your dispensers, if you're using a handheld type of dispenser, before that first generation moth flight begins in the spring. And it's important to understand this concept that mating disruption doesn't actually kill moths. You're not eliminating any moths in the sense of killing them from the system, but what you are doing is you're saturating the orchard with that female sex pheromone, and that's delaying or hopefully preventing as much mating as possible. And so research has shown that the way this works is that it's called false trail following. So you have out these dispensers, or little star-shaped objects on the tree there in the diagram, and the males actually end up going to those versus going to the female, and so then mating doesn't happen. So what happens is mating disruption over time will significantly lower the collie moth population in an orchard, but it's not magic and it doesn't happen overnight. So you can't just implement mating disruption whammo this year, you've never done it before, and expect complete control. It's going to be, you're going to need to use some supplemental insecticides, especially as you, transition into this, probably maybe a full program of insecticides at first until you get comfortable with it, your populations go down, and then you can transition into a more dependent on mating disruption. Also, we strongly recommend that you always run an effective monitoring program. So you're still running traps out there. And I'm not going to go into a lot of detail today about different kinds of trap lures. I can certainly be happy to answer questions about that, but again, I was trying to make this more of an introductory program information. And to mention that mating disruption is organically certified, OMRI certified, so for those of you that are interested in that, it's a good fit. So there are many different kinds of mating disruption products, and Marion introduced this, so I won't belabor it, but 3 of the major brands that we have the most familiarity with here in the Intermountain West are from Pacific Biocontrol. Their brand is called Isomate, and there's different kinds of handheld dispensers. They also have a mist type release, so like what we call a puffer or a mist release station. Sutera is another company, and their products are called Checkmate, and they've had a long-standing puffer, the one in the middle on the middle row there, where it's a little type of canister that has a bottle, aerosol can of pheromone, and it releases a puff, and you can set it to time it when collie moth is active, which is from just pre-dusk to about 10 p.m. at night, so you can have the pheromone releasing then and not having to have it go out all day, save some money. Also, they have products that are sprayable. You can spray them out onto the trees just like any kind of insecticide spray. And the last company is Trace. They've come along with some of their newer products that now have the Sidetrack product, and this Sidetrack, this has both the female sex pheromone, plus additional compound called DA, which is an ester that was extracted from ripening pear fruits. So this is a chemical that also attracts the female as well. And so their products, the sidetrack products, contain both the pheromone and the pear ester. And so they have handheld dispenser types as well as sprayable formulations.
Question about this sprayable, the Sutera. Is that similar to the GF120 that you use on cherries? And where are you covering the whole tree, or are you just going into the head?
So the question was on the sprayable formulations of the Codling moth pheromone. Is this similar to the cherry fruit fly product called GF120? And the answer is no. And the difference is that GF120 is a bait along with a very low level of insecticide. So what you're doing with the GF120 is you don't need to get the complete coverage because you're just putting out a band or a spray pattern of droplets that has the bait, the food bait, where the flies come to it, eat it. When they ingest it, the low level of the insecticide kills them. This is acting similar to the disruptant devices in that you've got to get a lot of droplets out there to release the pheromone and disrupt the mating behavior. So you're getting more of a full spray coverage with the pheromones. So it's differently applied, yes.
So is that pear pheromone?
It's not a pheromone, it's a chemical from the pear fruit, yes.
So why are you wanting to attract the females in there too?
So the idea being that if you're disrupting the females as well, so they're, instead of being as active about putting out their own pheromone and trying to call males, they're being disrupted as well, that you might get a little benefit kick from it. I have not seen any data, so this is a question for those that are online, to go back to the sidetrack products. It shows that they work better than pheromone disruption systems based just on the pheromone. I think they work, but do they work better? I haven't seen any data like that. So it's just their little gimmick or their little thing to add. So dispensers, that's been the primary tool that we have used here. It certainly was the first type of pheromone dispenser device to come along. The thought has been that the more release sources you have out there, so the more individual spots with pheromone coming out, the better you're going to do at disrupting the mating behavior, because you have more of these dispensers where the males are going to be going to thinking they're meeting a female and surprised they're not. And so they've traditionally been applied at 200 to 400 dispensers per acre. Now, if you're brand new to mating disruption, I would suggest that you go at the higher end of that level, because you've got to transition your orchard into that. If you've been doing disruption for a long time, what our growers have found is they can really drop off on that application rate and still have success because they're working with much lower populations. The cost is in that $50 to $100 per acre range, depending on how many you're putting out. This is a loop style, the CM Flex. You're going to always, you'll read the label and you'll see the products always say put a double or a heavier application on the borders. And the reason for that is you want to really make sure that you've got enough pheromone concentration around the borders of your orchard, that you're not getting problems with mated females coming in and that, having injuries show up on the borders. So the Checkmate style dispenser has a little clip on it. Again, their product is a 200 per acre application rate. They're sold in buckets of 750 per bucket, and their application cost is about $100 per acre. The Sidetrack, this is just the one without the DA in it. Doesn't matter, they look the same, but this one is just the CM. This also has a little puzzle piece, and you clip that over the branch of the limb branch. 200 per acre, again, similar price point. Yeah, Ron.
So do these have a shelf life, or do they have to be used to yearly purchasing?
So when you purchase them, you would hope that the company is sending you, fresh dispensers. They should be. You can store them in your freezer. Ideally, if you're keeping them frozen, they're not going to be losing any compound out. If you keep them in a cooler, that pretty chilled cooler, that should be pretty good too. If you have them just on the desk in your office at room temperature, they're going to be releasing pheromone. Now when you buy them, they typically come in some kind of sealed packaging, So that should be, you know, helping reduce that. But the best thing is to keep them in a freezer or as close to that as you can until you're ready to put them out. Okay? All right, so in terms of which dispenser is the best, they're all effective. So say that right up front. When we had some studies here in Utah a while back where we had growers that put out the different types, and Marion collected these and sent them in to have the amount of pheromone analyzed. With the Isomate Flex dispenser, they saw quite a constant release rate, so pretty good steady release rate of the pheromone throughout the first and second generation. So that's ideally what you would like to see. And so it was among the highest in release rate even after 140 days. And Dr. J. Bruner, thank you, I couldn't remember his name. He's retired now, so we've left our mind. Dr. J. Bruner did some studies where he looked at dispensers that had already been out. He looked at dispensers the second year. They were put out the previous year, and he was still able to find some release in that second year. So these Isomate dispensers release for a long time and a pretty constant release rate. The Checkmates, these tend to start off with a high release rate, and then that release rate drops down over time. And so these are generally a shorter lasting product, and overall were the least efficient in that they started off high and dropped down. The Sidetrack had the lowest release rate of the three over the course of the season, but it still was releasing at an adequate level to disrupt mating of codling moth. So in terms of timing, mentioned, so full bloom typically when you'd want to get these out, so you want to get them out before biofix occurs. You either use pheromone traps to set biofix, or we'll use our formula that we're going to talk about more. Eliminate the need for trap setting biofix. New orchard blocks, you definitely want to make sure you've got those double rates on the orchard borders, and you're going to need some supplemental sprays, so just plan on it. And you'll be needing to use traps to monitor through the season to know what to do. So Marion talked about numbers of traps needed. You always want more than one trap, because if a trap is in a bad spot, the lure isn't working, whatever it might be, and that's all you're basing everything on is that one trap and it's not working, then you're up a creek without a paddle, right? So you want to at least have two, and depending on the size of your blocks, you're going to want more. And so one trap per 10 acres is generally the recommended rate. We recommend that you use the CM-DA combo lure. And the reason for that is that we find better consistent catch with that. And there's now a new addition to that that we haven't really tested here in Utah, but it's adding acetic acid to that. And this is some research that was conducted in Washington, and found that the CM-DA lure with acetic acid added was even a better detector of codling moth in mating disrupted orchards. And the threshold that we developed here was that 10 or more, or when it exceeds 10, that's a threshold of when you need to be putting on a supplemental spray, or knowing that you're getting enough codling moth that you need to do some supplementation. This is some data that came from Helmut Riedel, who was at Oregon State University in Hood River for many, many years, and they looked at height of the trap. And there is an influence. So this is in meters on this graph, but the two bars in the middle that have the most moth catch, that's at 6 and 9 feet. So if you have your trap down here at 3, 4, 5 feet, you're not going to catch as many codling moth as if you have your trap at that 6 to 9 feet level. So it's important to place your trap high in the canopy. That's where the codling moths are most active in the evening hours when they're flying, and that's where you're going to get your best representation of what's going on. Yeah, Ron. So Ron asked if the size of the tree makes a difference. Certainly the canopy structure and the size will make a difference. This was done on a number of different tree sizes, and generally that 6 to 9 feet was the best height to get the most. You could probably get even more up higher, but it wasn't necessary. I mean, you got more up, it was similar up higher. You could see a similar trend. Here in this particular example, it dropped off when they got up to like 12 feet, but that might have been right at the top of the tree. And so if you get a lot of sun beat on your trap all day long, you're losing more pheromone, and so you don't have that benefit of the shading to keep the pheromone active. Okay, so I think, well, before I get to these newer questions, I'm going to talk about management, and then I'll come back to see if there was anything I didn't address. So beyond mating disruption, insecticides are the other key tool that we use for mating for codling moth. And as we've talked about already, the timing is so critical because we need to time it with that egg hatch and target those newly hatched larvae before they get into the fruit. Okay? So other things you can do, I mentioned the sanitation is so important. You don't want to leave your bins out in the orchard or any other kind of structures, boards, whatever it might be that codling moth will like to spin up and make a cocoon in. You want to have a clean orchard. Biological control is generally fairly minimal because once that larva is inside the fruit, it's protected. I won't say that there isn't some overall background biological control, but it's not a key way that we focus in on managing codling moth. It's mostly mating disruption and insecticides. So timing. So as you know, Marion puts out the advisories. Hopefully you know that. If you don't know about advisories, you need to definitely get signed up for those. But we also have this website, and I'll show you the app in just a moment, called TRAPS. So Temperature Resource and Alert for Pests. And this is a collaborative endeavor with the Utah Climate Center at Utah State University. And so if you've ever gone into TRAPS, in this particular example, it's a little small, I know, but I've picked all red orchards in Payson as the example. This is from last year, and I'm showing these little blue indicators on the far right of that table on the web page there show timing of key management activities. So I mentioned to you our traditional timing for application of insecticides for first generation is 220 degree days after biofix, because that's when the eggs start to hatch. Okay, so that's what that is telling you. And you can go in there and look at that yourself. Marion also tells you about that in the advisories. Now I want to tell you about something else that Marion has been informing you about for many years, but I don't know how many of you have been doing this. And this is an approach that they're really pushing in Washington, again, based on Dr. Jay Bruner's research that showed that, you know, in the spring, mating disruption doesn't work quite as well because it's cool. And so not as much pheromone is getting released from those dispensers in the evening hours when the moths are flying. And so what he found was a program where you put oil on at 200 degree days. So that's before they start to hatch. And what you're doing with that oil is you're suffocating all the eggs that are already out there. You get good coverage, and you're suffocating all those eggs by covering them with oil, and they can't breathe. And inside, the larva dies. And then you wait until 350 degree days. And what this corresponds is to when the egg hatch really takes off. So when it starts, egg hatch is kind of gradual, and then it accelerates. And that 350 degree day timing is right when the egg hatch is just really accelerating. So you've killed all the eggs that were laid up to 200 degree days. You wait 150 degree days more, then you put on your larvicide. That's the insecticide that kills the larvae when they hatch out of the eggs. And then you come back with another application if needed, 14 days later or whatever it is based on the product protection interval of what you're using, the insecticide that you're using. And so this is the program that they've been really pushing and finding that growers have really liked, and it's working better for them in Eastern Washington. And so our TRAPS table also has that information. So Marion has it set up where it tells you these timings as well. The 200 for the oil, the 350 for the beginning of peak egg hatch. Also, all of this can be accessed in the Utah TRAPS app, which is a free download for any kind of phone you have. And it's basically the same as the website. You know, that's what apps are, is they're shortcuts to websites, making it simpler and easier to get to the info. And you pick the site that you want to have the info for. Kaysville, Payson, Santaquin, whatever. Okay, and then the simplest way to get this information is actually from Marion herself, because she you know, gathers all this info, distills it down to the essential things to make it easy for you to use. So you guys, you don't know how much value you get out of Marion Murray. She gives you more than you can imagine. But here is the advisory. If you don't already receive it, it's a free subscription. All you need is an email. You get the attached PDF document. So this is one from May 20-something last year. And so here's a table in there telling you the timing. So for Utah County, you can see American Fork, Genola, Payson. This was the timing on the left if you use the traditional 220-degree day timing, or on the right if you use the oil, and the delayed timing at 350 to hit that peak egg hatch. So all this info is in there for your location, comes every year with the updates, the new information. So make sure that you're accessing all these resources. In addition, there's our tree fruit production guide, and Marion brought some copies from last year's printed version, but on the website was the 2016 updated version with any new insecticide, new pesticide changes that have come along. And as I'll show you, this is on our intermountainfruit.org website. And here, for example, if you pick spray tables, I picked apple, codling moth, and then there's a drop-down menu of all the insecticides that are registered in Utah for use for management of codling moth. And so it's all right there, and it's broken out into those that are reduced risk or kind of, you know, softer kinds of products. If you're looking for that, there's organics in there if you're looking for that. So tons of information in these different guides, and there's a hard copy from last year, and Marion's and her students are currently updating the guide that will be printed again in 2017. So there'll be a hard copy again in 2017. So you have some more questions that I didn't answer today. Our fact sheets are available, and here's a table in this, our codling moth fact sheet, that shows you all these degree day timings. You know, the when to put out the traps, the 220 when eggs start hatching, or the 200 when you can suffocate with oil, the 220 when the eggs start hatching, and so on and so forth. And we also have a fact sheet on mating disruption. And for those of you that are a little higher level of practice, there's in this fact sheet, it talks about using those CMDA combo lures to monitor codling moth while you're disrupting. And you can even determine on the trap whether it's a male or female. And so if you start catching females, which is the abdomen on the right here, the little heart-shaped tip of the abdomen, that's a female, that's a danger sign. I'm getting females in my traps, uh-oh, my mating disruption is not working as I thought, or you know, I have too high of a population, I need to make sure I get right in there and get some sprays on. The male is on the left, he has like a clasper on the tip of his abdomen. Okay, and so you can sex them, you have your hand lens, you can look at them on the trap, you can do this, and we'd be happy to help provide some more training on this. But I'd say this is a little more advanced. It's probably not for the beginner. Okay, so questions. How do you deal with codling moth pressure from neighboring orchards using mating disruption? So as Marion talked earlier, that's tough, because if you have your 10-acre block and you're disrupting, but you're surrounded by orchards that aren't disrupting, the best thing you can do is try to convince them to also use mating disruption. If not, you're always going to have to be using some insecticides, and maybe your insecticides can be more focused on the borders over time, where you can keep the interior clean without so much insecticide, but you're always going to have something to kill those immigrating moths, because disruption does not kill, it only disrupts, okay? What pheromone dispensers work best? So as I mentioned, we feel pretty strongly that the CMDA lure is more effective under a mating disruption environment than what we call the 10X lure. And I know a lot of growers have used 10X. You'll catch more with the CMDA, and we use a threshold of 10. But even one female caught then that's the signal that you need to have some supplemental treatment. Are individual dispensers better than puffers? So there's not an easy answer to that. It depends on your location, so the isolation from surrounding places, sources, the size of your orchard, how long you've been using disruption. I think puffers are a good fit where you've got more isolation, so less outside pressure, where you can have just one puffer per acre or two per acre, whatever the rate is that you're using. But you're not going to necessarily have as strong of a protection around the borders. And so I think handheld dispensers are more effective when you've got higher population stress in your orchard. Other questions on codling moth? Boy, I've already used a lot of time, so I'm going to fly through the last two. Not that I'll fly through twig borer a bit, because it's a lot of the same. Sorry, but I wanted to make sure I gave you the basics. So twig borer is the primary. What time did you want to be done, Marion? 11:30? Okay, 15 more minutes? Okay. Is that what you want? Okay. So peach twig borer is the major pest of peach, nectarine, apricot. A key thing to know about this, in contrast to codling moth, is that the larvae overwinter in these little cells. We call them hibernacula. They spin up some webbing and they make a little cell right there on the limbs of the peach. And so dormant oil and your insecticide is very effective on peach twig borer, and that should be your first line of defense, killing those overwintering twig borer larvae on the tree so that when they emerge and come out and start feeding on the new twigs, and so you're getting that tip feeding, that you don't have that problem, and plus you then just truncate the whole seasonal population. Otherwise, from bloom to petal fall, that's when the brown little caterpillars come out and start feeding in the shoots. And so you'd want 2 BT sprays or a spinosad spray timed at bloom, if that's another alternative you can use, and that's a program that a lot of organic growers use. And then in the second and third generation, this insect gets into the fruit, usually right at the stem end that's right up against the limb of the tree. It's hard to see until you pull it away, and then there's a twig borer in there. So some more pictures about it, what the moth looks like. It's a tiny little moth, but there is a mating disruption program for twig borer as well. And so the products that you can use are the Checkmate, which is the little membrane type dispenser, or the Isomate, which is the twin tube. And both of these are effective, and the Checkmate tends to last only 90 days, whereas the twin tubed Isomate will last about 150, so about almost double. So there is some difference in the efficacy, but we'll talk about the programs and how you could use them differently. Just some slides to show you that when you use disruption, this is Marion's data on Farm A in 2010 and 2011, and Farm B in 2010 and 2011, you can see the numbers of moths caught with the blue bars when you use mating disruption, and the green bars when you didn't. So a lot more moths when you don't disrupt, right? But how does this relate to control? So this is for percent fruit injury. You can see again that where you use mating disruption, you lower the injury, but you notice that there's still some injury from twig borer. And the bottom line is that twig borer is a little bit harder to disrupt than is codling moth, and we'll talk about that. So also the difference in the two dispensers, they last different time periods. So she looked at some trials where she compared the two dispensers. Farm A didn't have any injury, period, whether they did disruption or not. Farm B had quite a bit, and the Isomate is in the light blue and the Checkmate in the dark blue, and she saw less injury with the Isomate than with the Checkmate. And Farm C, they were pretty similar. So peach twig borer is harder to disrupt successfully than codling moth. So it's very important that your farm size needs to be at least 8 to 10 acres. If you have a high population within the farm, a high pressure from neighboring farms, it's not going to probably work very well. And she talked about orchard dimensions, how it's important. The best orchard size is more square than elongate, because in the elongate orchard, you have lots of borders, and the more chances for mated moths to come in. And the bottom line on why twig borer is harder to disrupt is that at distance, they use the pheromone. When it gets closer, the female actually makes a sound, a call, that attracts the male to her at short distance. And our dispensers obviously don't have that call. And so that's why it doesn't work as well. So in general, for twig borer overall program, that delayed dormant oil plus insecticide is so important to kill the overwintering larvae on the tree. And then using dispensers, Isomate or Checkmate, they both provided pretty good fruit and shoot strike control. With Checkmate, because it's only releasing well for about 90 days, you want to apply your first cover spray based on the biofix and the timing of when the model tells you that you should be putting your insecticide on to protect your fruit. And then you wait to hang your dispensers until about June 15th. And the reason for that is that you're now pushing back the time of the dispensers out there, so you're going to get that 90 days efficacy from them beyond that June 15th. Whereas with the Isomate, it's important to hang them early, in fact, really early. And they don't, you've got lots of pheromone in there, and it's going to be coming out for many days, and so hang it well before first biofix. Even if you're doing other activities early, you can do it early, because it's not putting out much pheromone in the spring when it's cool anyway. And so that's two different approaches depending on which dispenser you're using. So timing, so get that pheromone trap for biofix. And as Mary mentions, it can be fairly wide-ranging, anywhere from May 3rd to June 3rd. She's seen with the trap setting that she's done. In new orchards, make sure you have double the rate on the borders, and you put on supplemental sprays for at least 2 seasons before you start, counting on the disruption being enough. And again, with traps and thresholds, we don't have any mega lures for the peach twig borer. And so if you see 3 shoot strikes per tree or more, or you get 10 or more moths in a trap per week, those are thresholds that we've used to guide us that you definitely need some supplemental sprays. As far as more online resources, fact sheets, so one on twig borer, one on disruption specifics, oops, and I thought I had a slide in there, but I didn't, about insecticide recommendations are in the pesticide, or in the Intermountain production guide. So go to the peach section, pick on twig borer, and you can see the insecticides that are registered there for twig borer management. But mating disruption can be a very integral part of that. So any questions on twig borer management? Okay, so let's talk about the last key pest, which is cherry fruit fly. A big one for a lot of you, most of you grow cherries. So this one overwinters a little differently. It's a fly now, not a moth. The first 2 we talked about were moths. This one's a fly, and its larva or immature stage we call a maggot. And it overwinters as pupae, these little round, elongate but very hard-shelled pupae in the soil. And so that's where it spends a lot of the time of the year in the soil, and then comes out when we start getting into warmer weather. So the larvae feed inside the flesh of both sweet and tart cherries, and the females will start to be attracted to laying eggs on the fruit when it gets a little bit salmon or blush color. So as you know, when fruits start forming, the cherries are hard green, and that's too tough for the female to get her ovipositor into. And she lays the egg, in this case, directly into the fruit, so under the skin, not on the outside. And so that fruit needs to be soft enough for her to lay her eggs in it. So in terms of management, we're primarily talking about chemicals. And so GF120 was brought up earlier, so that is certainly one of the approaches, using a bait spray. But in terms of cultural controls, we did some studies where we found that any kind of barrier underneath the trees would disrupt the ability of the fly to get into the soil. And so on a small scale, landscape fabric can be a good fit. Also, any kind of dense vegetation was much had much fewer fruit fly pupae in the soil than just open bare soil, which had the most. There's many different chemicals, and again, you can refer to the Intermountain Fruit Guide to get a full list. But I've listed some of the primary products. There's a number of different modes of action that we use there. Also predation by chickens, guinea fowl, anything like that. If you have an orchard where you can run chickens through after the cherry season, after the cherries are picked, they're great for picking up cherry fruit fly larvae and pupae. So monitoring is important because you need to know what your population pressure is and when they're active. And so Marian introduced the yellow sticky panels. And these are our key traps. She mentioned that having an ammonium bait, and I'll show you a slide in just a minute with some data that shows you 6-7 times more flies caught when you have an ammonium bait. We actually use ammonium carbonate, which is a little more stable than ammonium acetate, but both of them work just fine. We've evaluated different kinds of traps, including some 3D traps, but the bottom line is probably the panel trap is the most efficient, easiest to use. I've got pictures of 3 kinds of panel traps. They might all look the same to you from the outside. They're yellow sticky cards. The one on the left is our standard called Pheromones. This is a cardboard, cardstock-based trap. We've been using it since before I came here, and I've been here 27 years. When you get it, it's folded inside, you invert it, and the sticky side, yellow sticky side, is out. In the other 2 traps in the middle and on the right are made out of plastic, and these are translucent. So in this case, the light actually passes through. And so these have a different kind of tacky surface. It's put on with like an iron, almost like a hot press kind of tackiness. And so it's a little different. Instead of being that goopy adhesive, it's a sticky, tacky adhesive. And the reason why I looked at translucent traps is that In the tree there, the little blue things are supposed to be the cherry fruit flies. When they spend most of their time on the inside of the canopy of the tree during the day, hanging out, waiting, males looking for males actually sit and wait for females, and then they pounce on them. Females looking for fruit that has salmon or more red on it to lay her eggs in. But when you hang that trap up in that cherry tree, when you have the cardboard trap, what happens is it becomes just a dark structure to the fly inside the canopy. The fly does not see any yellow color coming through the trap. It has to be almost out where the sunshine is shining on it to really see that yellow color. And yellow is attractive, visually attractive to the fly. Whereas if you've got the light passing through that trap, the fly sitting here on the inside of the tree canopy, it sees that yellow and is more attracted to the trap. So the translucent traps can increase trap catch. So this is just some data from last year, and I've been looking at these for a number of years. But on the upper slide, the blue bar represents that yellow sticky strip trap, which is the most translucent, and we caught the most flies on it. The one in the middle is the Pheromones, and the one on the left is the Alpha Sense, which is also translucent. This year's trial, we didn't see a significant increase with the Alpha Sense over the Pheromones. I have seen that in past years. The Alpha Sense is not quite as translucent as the yellow sticky strip. The graph on the right shows you what happens when you add ammonium bait. So a total of 20 flies on a trap in the yellow bar there where there was no ammonium carbonate, and almost 140 flies on the trap when it was with ammonium bait. So almost a 7 times increase in numbers of flies caught when you've got the ammonia. So it makes a big difference. Some downsides of the yellow sticky strip are that it's lighter weight. And this year we had a lot of wind when we were running the trial. It flopped in the wind a lot. And some of the tie holes that we had to tie it in the tree, they actually ripped out. So that one is still, I'd say it needs some work on it in terms of making it, ready for prime time, so to speak, in the cherry orchard. The Alpha Sense traps is heavier, and I probably flapped a bit more in the wind, but it didn't tear out and it held up just fine. Although this year we didn't catch any more on it than we did the Pheromones, but we did in 2015 when we tested them. So some concluding comments about cherry fruit fly control, besides just telling you use insecticides. There's some concerns with its management. A primary source of cherry fruit flies for growers that are doing a good job of treating their cherry orchards and keeping their fruit fly populations down, a primary source can be the external, so outside your orchard. So if there's some backyard cherry trees or feral trees that aren't being treated, when these females, we call them gravid, they already have mature eggs in them, fly into the cherry orchard, they're especially going to be infesting probably those border trees because that's where they're going to encounter first. But the challenge is there's no delay in the time between when they get to your orchard and when they can lay eggs. Because if she's already mature, whammo, she can lay eggs right away. And so how fast does your insecticide need to work to kill that fruit fly? Very, very fast, right? And so in some lab studies, we actually found that only 2 to 3 hours if that fly was put on a ripe fruit, within 2 to 3 hours, there were eggs laid. And so it's not very long. And so that's a real challenge if you've got outside sources. The other sources would be more internal, like if you've got a hot spot that built up last year where for some reason the spray coverage was poor or something like that, then you may have a hot spot within your orchard of some flies that are going to be coming out this year. So at the annual Hort meetings for many years now, I've been talking about some other technology. Started by talking about GF120 when it came out, and it's a bait spray. So it adds a feeding attractant to it, so to get the female to want to eat it and kill her before she has a chance to lay eggs. The other one on the upper bullet above that one are the systemics. And so there's a class of insecticides now called the diamides. And there are now actually 3 of these that are registered on cherry. So Altacor is the oldest one, which is okay. It works pretty good. It's actually quite good for codling moth and leaf rollers in apple. But the second one listed there, Exaril, looks very, very good on cherry. It has a little bit more activity, a little more mobility within the plant. And so this is systemic, and what's happening is you spray it on the tree, it's being taken up, it's actually getting into the fruit, and it's killing the eggs as they hatch and the larvae as they start to get active, if there are any larvae that are already there in the fruit. A new one that's coming out this next year that I've also evaluated and looks superb is going to be called Harvanta. You haven't heard of it yet because it hasn't been registered yet, but it should be registered this coming year. And so these all, and they have some broad activity. I've listed the other kinds of insects that they also work against, so they're not just fruit fly products. So again, we have our fact sheets and other online resources. You can go and look up specific insecticides and longevity of those products and how to fit them into your program on our Intermountain Tree Fruit Guide. Okay, so I'm about out of time, but I just want to real quickly give you a very brief update on two of our invasive pests. And for some reason, the title on the slide, Brown Marmorated Stink Bug, disappeared when the slide was altered in some way. But anyway, this is brown marmorated stink bug. Many of you have heard us talk about it for many years. We did start picking up, not we, some of the growers that are here in this room have started picking up some more in this area and around places where fruit is being grown. So obviously we are concerned about it. To date, there has not been any economic damage, so no feeding injury detected, reported, that we've observed in fruit or any other agricultural crop. But there are enough of these guys now that they have now become a nuisance pest in Salt Lake City, so particularly around the University of Utah campus, the avenues, and other places in Salt Lake. And the reason for that is that the adults like to get into protected sites to spend the winter. So right now we've been getting lots of pictures coming in, people saying, hey, I found this bug in my house. What is it? Well, it's a brown marmorated stink bug. And so they are becoming more of a nuisance pest. Catalpa tree, which is, maybe you can tell, shown in this picture, they have the long legume pods on them. That's been the key tree that where we've been finding reproducing populations of this. And this is Dr. Lori Spears. She's talked with you many times. This is her information. And so if you go to a catalpa tree in Salt Lake City, a good majority of them have brown marmorated stink bug on them. And they're laying eggs there and they're reproducing there. In this map, you can see where brown marmorated is distributed across the country. We want to get Utah changed to yellow, which means we have nuisance level, and it's established here, and we have nuisance level population, so we've asked for that change. The best way Lori's been able to find them is by beating the bushes, literally, beating trays to look on different kinds of plants and find them there. It's a little hard to see in the blue text, but the traps have not worked very well. We have not seen very many adults caught in traps, and so we're concerned about that. We don't have a good monitoring tool. Lori and I have just hired a new graduate student named Cody, and he's going to be looking at brown marmorated stink bug, different kinds, and looking in the urban and the agriculture areas to see where we can find it on which kinds of plants, and the timing of when the adults and the immatures are active, etc. And also looking for if there's some biological control going on, because there's a lot of excitement with that across the country on more biocontrol. Cody has a colony of them now. I know those of you think that's strange, but if you don't have bugs to work with, it's hard to do the research. So we have a colony. We keep them very close to us. We don't bring them to you. And those are the eggs. That's an egg cluster laid there on the side of the cage. Okay, spotted wing Drosophila. So this is one that's been around since 2010 here in Utah. As I'll show you in just a moment, it really hasn't yet become a threat, so that's a good thing. And I'm hoping that this one will not become what it has become in more southern states, warmer places like North Carolina, Georgia. They're dealing with this in a big way. But even Eastern Washington is having some challenges with this. Another Asian pest. All our new introductions seem to come from Asia now. We share around the world. It likes ripening fruit, but it can get into fruit before it's totally ripe. And this kind of fruit fly is like, it's related to the ones that you put your banana out on the kitchen counter and it gets ripe and you get those little tiny fruit flies in it. That's what this one is related to that. But it's a particular species that will attack fruit before it is ripe. And this is a picture of some cherries in Washington that were attacked by spotted wing. So not a pretty sight. There's a distribution, pretty widespread now. You can see the counties that it's been detected here in Utah. This is just some data showing you through last year. It doesn't have this year's data because Lori's still collecting it. You can see our numbers are going up. That total number of spotted wing. We've got more traps out, so that's a big part of it. We're looking more as once we found it, but also our numbers have been increasing. This graph shows you that that gray bar is the temperature range where spotted wing is ideal. So it doesn't like it too cold and it doesn't like it too hot. Well, Utah has a lot of hot and cold, and so we are not an ideal place for this fly, for this pest. So those little squiggly lines show you what would be predicted in terms of when the spotted winged Drosophila would be out. The stars show you when she actually starts detecting them. And you notice that all the stars are for the most part, except for one exception, one catch early, they're all late. And so what happens is we don't see these until August, September, October. Actually, here peak catches come in November, right now, before we get too cold. And so in terms of what's going to be most at risk from this insect, it's going to be those late maturing fruit. It's not going to be our sweet cherries and other things early on. It's going to be the female has a little ovipositor that she can cut into the fruit. And so it's going to be our, I'm going to skip that. It's going to be our, I thought I had it in there somewhere. It's going to be our late season fruit. It's going to be our fall raspberries, our late maturing peaches. But so far, we really haven't seen it pick up and move into orchards and become a big deal. She can catch lots of them, like in my backyard. She can catch tons of them at the mouth of Santaquin Canyon and all those wild fruits, but she don't really catch or don't see any damage in the commercial orchards. So anyways, just some cultural management. I wanted to remind you that on the back table, we brought copies of the invasive fruit guide. So for brown marmorated, spotted wing, and these others that you see on the list, they are a lot of detail, a lot of nice illustrations that Cami Cannon, I don't know if we introduced her. She's sitting at the back table and helped you sign in when you got here. She helped us put this guide together. So this is a great resource for you to look up more detail. So that's all that I have. Any other questions?